Sciences at the University of Montana. He and his students, I love that he includes his students on everything. So important to me.
They include me. Actually,
okay, well, good. It's good that they let you. They study community ecology with an emphasis on exotic plant invasion. Specifically, they're interested in how organisms interact with each other because the interactions can sometimes shed light on the fundamental aspect of communities. He and the students have developed strong collaborative relations recently in the last couple of years with Brazilian researchers. I think that's really smart to have your collaborations be in cool countries that you can go to. In 2013, he was named the University of Montana undergraduate mentoring, or given the undergraduate mentoring award, which again is close to my heart and some of the others in the room. So congratulations on that. And he is our new EPSCoR director. So Ray Callaway.
Thanks, you guys, for coming to listen to this. I was here in October of 2012, just a year ago. So some of you guys have a very high pain threshold, and I do appreciate it, though, especially you, Chad. This is new stuff, though. So, well, not all of it, but most of what I'm going to talk about today, I didn't talk about last year. So hopefully it'll stimulate some new ideas. So Martha didn't mention it directly, but I'm just to let you guys know where I'm coming from. I'm a community ecologist. What I know most about is plants. And so I work at that scale, kind of the scale of organisms interacting with each other, whether it's plants and plants or insects, or plants and soil microbes and so on. That's what I do. That's sort of the scale I operate at as a scientist. But I want to at least attempt to put my work, my talk, at this scale, great big scale of the Earth. And when I mean this scale, I don't mean like, Steve Running or other folks here that like, do big models and, satellites and NASA and stuff. I mean this kind of scale that these boys thought about, 150 years ago. So Wallace and Darwin. And as you guys know, basically they hung around in the jungle, sailed around the world on boats and made observations. They didn't do statistics or collect data. But their observations at the scale of biogeography, the scale of the world, have completely revolutionized what we know about biology. This was it. And basically, the most fundamental way to think about this is that they observed that when organisms were isolated for what appeared to be a long period of time, that they diverged, they evolved through natural selection. And that's how they came up with the theory of natural selection and evolution, by simply making those kinds of biogeographic observations. And as an outcome of this, I mean, we all know now that this kind of geographic isolation has led to, really by far the diversity on Earth that we understand, or that we can count and measure and study as ecologists. And this is true and widely accepted to be true for just about every taxa, every form of organism, except for really one. And this is in transition, and some of the people in this room have actually played a big role in this, maybe not with soil biota, but with microbes in general and their biogeography. There's been debate and there's controversy over whether or not organisms that live in this stuff really do show biogeographical patterns that are meaningful. In other words, if you take some soil from Africa and you take some soil from Montana, you're going to get some very, very similar ecosystem processes. You're going to get nitrogen fixation probably. You're going to get nitrification processes going on. You're going to find a lot of the same basic ecosystem processes going on. And so for a long time, in part influenced by a German, Bas Becking, at least I think he was German, or Dutch, the general way of looking at things is that, microbes, bacteria, soil biota don't really show that much of an important biogeography, like the rest of life on Earth. So that's kind of one kind of theme I'd like you guys to keep in the back of your mind as I go through my talk, and to kind of tackle or ask questions about biogeography and these kinds of organisms. I'm going to use these kinds of organisms. Okay, and this is what the sort of thing that my lab does for about 75% of my research effort. So these are invasive organisms. These are all organisms that have been moved from one part of the world to another. And when they arrive in their new range, they do seem to do something really very different ecologically. They become much more abundant. They have much greater impacts, and they show some kind of an ecological shift that can be pretty impressive. Now, these organisms come from just about every imaginable taxa, but I'm going to focus on plants, mainly because this is what I know about. But these are all examples, again, I'll be trying to be real clear about it, of what I would call an exotic invader. And a plant that humans have moved from one part of the world to another, and once they've arrived, they do something really different. And typically, they get far more abundant in their non-native range. Okay, and again, they have much greater per capita and sometimes not per capita impacts on native species. The other reason why it's kind of nice to focus on plants is all plants, or almost all plants, have a very intimate relationship with biota in the soil. So the interactions between these plants and soil biota provide us an opportunity to ask questions about biogeography. And in that case, of course, evolution. Okay, evolutionary trajectories in different parts of the world. So, an example of various critters that can find in the soil. Most of the talk, not all of it, I'm going to treat the kind of soil as sort of an almost black box. And I want to start off just for background for people that haven't really thought much about this. There's a lot of different ways that microbes can affect plants. I've got kind of a list in red around the box here. Everything's from either being pathogens, killing or hurting plants, competing with them for resources, providing mutualists like mycorrhizae or rhizobia for nitrogen fixation. There's a suite of different organisms in the soil that interact with plants in very, very different ways. And again, most of the time, I'm going to kind of look at this as one big, giant group. There's going to be one example in which I try to tease it apart in a kind of a reductionist way, but just as a little bit of a warning, it was completely unsuccessful. The success has been looking at the soil as a group, as a functioning community. So how do plants affect soil microbes? Well, they can compete for resources. Okay, they exude biochemicals, and through specific biochemistry, they can affect organisms in the soil. And they're, of course, a primary source of just fixed carbon, energy in the form of carbon that microbes can eat. So plants affect soil microbes affect plants. Nothing particularly unusual there. And generally, over the last 20 years, there's been a big sort of thrust to study the relationship in kind of a feedback sort of approach. And this is where I'm going to spend probably too much time. I'm going to beat a dead horse, I'm afraid. But I'm going to talk about these kinds of feedbacks, but I'm going to try to give it a biogeographic perspective. That's really my goal. And just to make sure we're all up to speed, got a couple of cartoons to show you what I'm talking about. These are the guys responsible really for bringing plant soil feedbacks into the world of biology. Jim Beaver in Indiana, Wim Vanderputten in Holland. And this cartoon shows an example of something that doesn't happen very often, but it would be a positive feedback, where you start off with individual plant one, okay, and you have kind of a mixture of positive and negative pathogens and mutualists. Positive feedback, well, it cultivates the mutualists, and you get a bigger plant. So the individual two planted in the same soil, the feedback is positive. Planted in soil where a conspecific is grown, you get a benefit for that. And you can see why that would be relatively rare, because this can't go on forever, obviously. That would be the only organism on Earth. So more common is this, a negative feedback. Okay? And I'm going to talk about both positive and negative and neutral feedbacks. But here you start with the same plus-minus kind of arrangements of mutualists and pathogens. Negative feedback, you cultivate the pathogens, that's the normal thing, and you get a smaller individual too. So just keep that in mind when I'm talking about feedbacks in the context of real experiments and so on. This is a negative feedback, positive feedback. So probably the most important, other than maybe discovering the idea of this feedback, the most important paper that's been published, and that starts to give kind of a hint of biogeographic differences in feedback, was published by this guy, John Coronomos, who's now at UBC Okanagan. This paper was published in 2002, and what this graph shows is the strength of the feedback over here. So these are positive feedbacks. These are negative feedbacks, all right? And then there's a kind of a list of species down here on the x-axis. All these species over here are showing positive to neutral. All of these are showing negative. And the cool thing is that all of these species over here that show these positive to neutral feedbacks are invasive. Invasive in the old fields that he studied in eastern Canada. All of the species that are showing the negative feedbacks are rare native species. right off the bat, there's a hint here that something evolutionary is going on. And if you're interested just in the process of exotic invasion, it's suggesting that these invaders are escaping something in their native range. I mean, it's not shown in this graph because these are only soils from Canada, right? But it would suggest that these guys are escaping some kind of density-dependent form of control that might allow them to go wild and actually act like an invader. So that is a very striking contrast between natives and invaders. John took it a little bit farther, and he explored this in the context of being a little bit reductionist. Using different filtrates, looked at the AMF, so this is these are mutualist fungi, and he looked at pathogen components of the soil. And what he found was that if you look at the mutualists, everybody seems to be able to get a mutualist that needs one. There's these positive across all of the invasive and rare species. But when he played around with the pathogen part of the filtrate, then he got the sign of his negative feedback and nothing over here for the invaders. Negative feedbacks for natives, density-dependent kinds of control, hinted from that. Nothing going on with invaders. So this pathogen fraction appears to be pretty important. And I believe that this would include both some forms of bacteria and but probably primarily fungi. So this aspect still is pretty black box. So for those of you who are ecologists, this pathogen bit plays a pretty big role in a really fundamental hypothesis, probably one that you would get asked on your oral comps exam. Plays a role in this classic Johnson-Connell hypothesis for mechanisms that can maintain diversity. And primarily the idea was maintaining diversity in tropical forests. So these two guys came up with kind of the same idea roughly about the same time in the 80s, I believe, or the late 70s, that density-dependent processes or distance-dependent processes involving pathogens could be a major driver of incredibly hyper-diverse places like this. Okay? And what their idea was, their idea is sort of illustrated in this cartoon. So over here's mom of theoretical mom. Seed dispersal near mom and a lot, far away from mom, not many. But mortality, or seed and seedling survival, is the hypothesis was. They didn't start off by collecting data. They started off by making a hypothesis that survival next to mom was going to be low because the enemies of mom are concentrated right here. That could be pathogens or herbivores or something else that mom might do to consumers. And so recruitment was going to be pushed away from mom and spreading out these conspecifics. And thus, of course, making room for other species and the potential for diversity, at the local level to go up. And this has been shown, supported a lot of times in papers studying in the tropics. Not everything, but it's a pretty reasonably well supported hypothesis. And that's pretty fundamental to ecology. So keep this in mind now in the context of where I'm going to go with invasion. It doesn't happen only in the tropics. This is a very cool paper published by Alyssa Packard and Keith Clay, in which they studied a North American deciduous forest tree, Prunus serotina, black cherry. And I'm only going to talk about it in words because I'm going to go on to the invasion stuff. And its relationship with a pretty well identified fungus. And for this tree, they found excellent evidence for this Jansen-Connell hypothesis. Seedlings near mom died off and were killed by Pythium far more, Pythium in the soil, far more than seedlings that were away from mom. So they established a really nice pattern of this Jansen-Connell kind of density, distant dependent sort of relationship. And the interesting thing about Prunus serotina is that this guy here, Kurt Reinhardt, realized that it's invasive. Not in North America. It's one of our natives. This is one of ours that we sent. And so he was my PhD student, but he did all of this stuff independently of me. And what he did was arrange a collaborative relationship with this guy, Wim van der Putten, I mentioned earlier. And he studied Prunus serotina in its native range and in its non-native range. And the first thing he did is ask, well, does it really look like an invasive? What does it do differently in its non-native range? And what he did was simply go out into the field, find a prunus serrata, find a tree, measure the distance between it and the next tree to get an idea of density. So if you look at the native range, that's these white bars here. These are trees, these are saplings. If you start off with one prunus serrata that you encounter, and you run your tape around until you find the next one, you got to go 40 or 50 meters away to find the next one in the native range. Okay, and there's evidence from Packer and Clay that that's because of density-dependent mortality. That's what we expect in natural, uninvaded communities. But if you go to Holland, where it's not native, okay, the density is far higher, many, order of magnitude higher. You go about 3 meters, and you come to the next tree. And so this is a great example of an invasive thing, okay, the spatial pattern. So then what Kurt did is he collected soil in these two ranges and experimented with that soil to ask if feedback effects were different in the native and non-native range. And as far as I know, this is kind of a goofy story. Kurt and I talked about this kind of stuff a lot, and I was playing around. I'll show you next. I was playing around with spotted knapweed, and Kurt was started to do this, and we talked about maybe let's collaborate on these things. And I said, well, no, let's compete. Let's try something different. Let's see who can get there first. You do your thing, and I won't be involved. We'll talk science, but let's just see who can publish first. Let's have a little fun. And he won. He published first, and I published second, so he gets to go first here. So anyway, he took soil from the native range and the non-native range, and he did some experiments. He planted seedlings at different densities. He got soil from under trees, away from trees, and then asked a few questions, whether or not you sterilize the soil or not. So if you look at the native range, this is mortality, lots of mortality. They're dying, and seedlings are dying in their native soil. They're not dying so much in their non-native soil. Interestingly, if you get a little dense collection of seedlings, this is 3 seedlings planted together, and you put them in soil that's from under bomb, mortality is 60%, really, really high until you sterilize, goes away. Okay, so that indicates there's something bad in the native range. Packer and Clay would have said, yeah, right on, we knew that. So none of that shows up in the non-native range. There's no effective sterilization, nothing. And it's even more dramatic if you come down here and you look at growth, whereas this column doesn't count because they're all dead, so forget that. But if you look at all of the other columns, sterilization increases the growth of the seedlings, okay, in the native range, soil from the native range. If you go to the non-native range, sterilization actually showed this is a positive effect, which is a little bit weird. Sterilization decreases the performance, the growth of the seedlings. So arrows going up, arrows going down over there suggests something kind of cool. It suggests something about biogeography, and that there is some real biogeography with these soil organisms, at least in the way that they participate in interactions with this particular plant. Suggests evolution, in other words, evolutionary relationships in the native range, and it suggests that maybe, in some cases, invasion can be either driven or exacerbated by release from these organisms. One would assume, co-evolved biota in the soil that control you. Okay, I'm going to move on to my defeated story in the race. Centaurea stevie, or spotted knapweed, is kind of one of my lab rat species. Got interested in this thing. You guys are all familiar with it. It's a good invader. It doesn't do this kind of near monoculture thing in its native range. It looks more like this. I've hunted all over Europe for this thing. It's a weedy thing in Europe, but it usually, as you can see in this picture, it grows with a bunch of other species. You don't usually find it, in big monocultures, maybe little tiny monocultures, but nothing like we see in Montana. It's a great invader. It shows this biogeographic pattern of big differences in abundance and in impact, which we've measured directly. So Centaurea stevie did the same thing that Kurt did, nothing special here. Went and collected a bunch of soils in Montana, collected soils in France, and began some work that actually, so this is published less than 10 years ago. I would have a hard time publishing this work now simply because the protocol and the way of doing these feedbacks has changed so much. We just simply sterilize the soil. There are problems with that. So we collected these soils, started growing and playing around with them with knapweed. Here's knapweed, little sickly-looking knapweeds growing in French soil, same seed source. Here they are growing in Montana soil, happy. That could be for any number of reasons. Fresh French soil, though, little kind of scrawny-looking Knapweeds, sterilized French soil. Oh, Montana looking knapweeds, suggesting that something might be going on. And again, sterilization has direct effects that could mask simple biota effects. And so we did this for a bunch of different, all those soil collected from a bunch of different places, different rhizospheres. And in general, you can see the basic pattern here. Sterilization always improved growth. But across a bunch of different sites, we found that sterilization had a much bigger release in the native range. Now remember, here knapweed's the native in Europe. I kind of switched continents where the invader was going. But here, so in Europe, native range soil get released if you sterilize the soil. And just to show you a little more detail, and also to kind of set up the slide after this, these are the different sites at which soil was collected. And so then I picked, it's kind of a cheesy setup, but I still did it this way. I picked soil that looked really nasty in France from the Central Massif, and soil that looked really good from Missoula. So in other words, here sterilization kind of hurt the thing, and here sterilization made it 10 times bigger. So I took those soils and did a feedback experiment, like I showed you with those pots in the cartoon, and got this pattern. And again, this shouldn't be any surprise. This is kind of what I showed you for Kurt, and it's what you would also expect from the data I showed you just a minute ago. Here's French soil, trained by Centaria, or trained by a native French festuca. Oh, I know, we have it here now too, but it's native to France. And what we found was that knapweed did not like growing in soil that another knapweed had previously grown in. There's a difference here, and if you sterilize it, it goes away. Okay, suggesting, again, just negative feedback at home. Montana soil showed the opposite, a positive feedback. It liked to grow in soil where a Centaria had grown before, and sterilization made it go away. So it's this contrast to this contrast, again, that suggests biogeography, evolutionary history, and that it matters. And that maybe escape from these critters in the soil play some role in spotted knapweed's invasive success. So I'm going to stay kind of in the same theme. I'm going to keep showing you some data that are related to feedbacks, but this is I'm going to show you my effort to kind of tease apart some of the black box. And this is what I said was right off the bat was kind of unsuccessful. So I'm going to make this story pretty short, I think. We pulled apart again with colleagues, focused in on particular pathogens, and we focused in on mycorrhizae from the different ranges. And we focused in on mutualistic rhizobia from different ranges for an invasive species, actually not invasive here, but invasive in Europe. This is black locust, or Robinia pseudoacacia. It's a kind of a weedy tree in our eastern forests. It's been spread all over the world, and in some parts of the world, it's considered to be invasive. So this is its native range, the Great Smoky Mountains, and also in the Ozarks. This is where historically the range is thought to be limited. Now all over North America, and it's all over Europe. It's supposedly the most common tree in Hungary. So, just like these other stories I told, went out, collected soils across a pretty wide portion of the invasive range, collected soils right from the true native range, at least that's what we thought we could do. And then we kind of, just for the heck of it, collected soils across this expanded range as well, and tried to do some reductionist science. And again, I'm going to try to summarize this reasonably quickly. Grew larger in soils from the invasive range, good. Nodule production didn't differ among ranges, that's interesting. It's picking up mutualists wherever it wants. I'll get to that again in just a minute. But we didn't find this target pathogen, and there's a lot of reasons to target Fusarium. In other words, Fusarium shows many different strains in different places in different soils. So this Fusarium from the native range, and Fusarium, this is a bad soil fungal pathogen, and Fusarium from the non-native range didn't have any difference in effect. It had negative effects. We couldn't tie it into this particular pathogen. Okay, that from the literature we would have guessed should have been pretty bad. So right off the bat here, we're not really seeing a lot from being reductionists. Interesting, sort of a little bit of an aside. Robinia, again, this invader has just about everything you could imagine as nitrogen fixers in its nodules. It picks up nodules from a very wide phylogenetic range. And it doesn't seem to be limited in any way at all. So again, there's no real story here. We also tried to be reductionist by breaking apart the soil biota community into a few different groups, and this is really just done by filtering, so it's pretty loose. This isn't loose, AMF you can pick out. But these are just simply different fractions. And I wanted to show you we did this. Now I'm going to blow up 2 of these panels to try to be a little bit more explanatory. We found out something interesting about NSF, about the AMF, but that didn't explain invasion. As far as I know, this is the first time anybody has shown that a plant really likes AMF better from its home range. So the feedback effect was very positive if we used AMF from the Smokies or from the Ozarks. And then really very, very weak effects and tending towards negative for its expanded and exotic range. So that's kind of cool, but it doesn't explain invasion.
What's AMF?
Arbuscular mycorrhizal fungi, so the mycorrhizae. But if we look at the soil in this holistic kind of way, we do get something that fits, just like I showed you before. There's nothing new here. In the North American native range, the strong negative effects existed, just like before. And again, we had to look at the soil as one big chunk. And we had weak negative effects when we got outside of the native range. This has the potential, at least, it correlates or corresponds with invasion. I wouldn't have expected this, but it struck me as kind of interesting that we really did need to keep the soil biota as intact as a community as possible. And what it suggests is that there are interactions among all the gajillions of organisms that live in here that are important to the total outcome, which actually made it hard to publish this paper. We got criticized for saying that, reductionism really failed in this case, and we had to be holistic about looking at the soil. So I would say embrace the black box, at least in terms of invasions and soil feedbacks. One other thing before I move on, just a plug for this plant soil feedback thing. This guy in the hole is Andrew Kumatiski. He's at Utah State, and he's very good sort of invasion soil biota type person. And in 2008, he published a meta-analysis in which he collected as much information he could about feedbacks and the nativity of species. This diamond here, these are invasive species, and that's an effect size for a whole bunch of species, and it's weak. It's close to 0. Okay, this is the effect size for native species, and it's quite strong. It's getting down here close to -0.1. So again, I guess what I would say, if for any of the many hypotheses for invasion, exotic and plant invasion, this soil biota suppression business is probably the most consistent that I know of in the literature. I mean, you probably couldn't find this sort of consistency for pretty much any other hypothesis out there for why invaders do their invasive thing. Okay, so all of this been focusing on feedbacks. What I want to do now is kind of give you a couple of little vignettes in which I take away that arrow and just think about an invasive plant on soil biota, again, sticking with the soil biota theme, and sticking with the biogeography theme. This example I have presented here, so sorry if it's going to be repetitive for a few of you. This is work that I did maybe 2005 to 2008 with an invader in the Midwest, and it's also in the Northeast, called garlic mustard. I'm not that familiar myself with its natural history. I don't live there, but it seems to be a pretty good invader, doing this kind of thing in the non-native range and being a lot less dense in its native range in Europe. This isn't my work, but it sets it up really very nicely. This was done by Kathy Stinson, and what she did is found that soil on which garlic mustard had grown was a terrible place for mycorrhizae. So these two y-axes here show some characteristic of the ability of the soil to provide this mutualism with mycorrhizal fungus. This is garlic mustard-free soil. It's great at providing mycorrhizae to these native species, but if you collect soil from under garlic mustard, it sucks. You can't use that soil to provide inoculum for the native trees. That's pretty interesting. That's not too surprising. Alliaria is a mustard. Okay, so it's not mycorrhizal. No mustards are mycorrhizal as far as I know. But the fact that it really kills and suppresses mycorrhizae in our soil is kind of interesting. And then I got into the act by, again, by turning this into something biogeographic. Soils collected in both ranges, and then sort of a big science by siege experiment comparing the two ranges. Okay, and so what we did, we collected soil from Europe and North America. You're trained it or not by the garlic mustard. That's it. And then we grew a lot of different species in trained and untrained soil, and species from the same places we collected the soil. So we're just simply asking, do you see that same garlic mustard effect in its native range? This is the best slide of the 3 or 4 I'm going to show you. This is this mycorrhization potential. How good is the soil at providing mycorrhizae? This is what we found, and it's exactly what Stinson and all found. So if you train the soil, it's very poor. North American soil, it's very poor at providing mycorrhizae. But the same thing doesn't happen in Europe. In Europe, garlic mustard isn't killing the mycorrhizae. Could leave it at that, but we actually tried to play around with the ramifications of this for species that grew in the same sort of communities. So these are results from our two sites in, at least for this experiment, in Europe. And if you look across, these are all just different species. If you look across the effects, this means a positive effect of Alliaria. This means a negative effect of Alliaria. If you look across those lines, you see mostly neutral, couple of positives and a negative. Not much going on. And here's the mean right here, pretty much 0. So species that need am fungi, need their mutualists, in Europe are not affected by Alliaria's effect on the soil. But it was different in North America. Now you see a whole bunch of negatives across here. And here's the mean effect on emergence. Okay, so in this particular case, we see a biogeographic effect again. And it appears to be, at least this corresponds with being moderated through this one component of the soil biota. So this is emergence, and again, I should emphasize, these are for species that we measured as needing am fungi, as doing much better with the mutualists. We excluded species from this graph that don't need am fungi. I'll show you that in just a minute. A little simpler graph. This is total biomass over here, so now it's how big the plants get, how they're growing. And this is the control, Alliaria trained across all 4 of these sites in North America. If you train soil with Alliaria, these particular species do a lot more poorly. And we also know that these particular soils don't have the potential to provide the mutualism. And in Europe, we didn't see that at all. This positive effect is really driven by one species. It's a little bit of a fake out. But it's pretty neutral in the native range. And again, this biogeography is what I'm talking about. In Europe, it would appear that am fungi get along just fine, have evolved to get along just fine with this invader, whereas here, our am fungi get killed. This is a quick slide to show you guys that for non-am species, these now are the few species that we had from these sites that didn't improve their growth when they were infected with am fungi. Okay, and in this case, none of the non-am species showed any negative effect of soil being trained by garlic mustard. So from this, other people have argued that garlic mustard is allelopathic, and they have evidence for it. We didn't find any evidence for that. We found that garlic mustard was antimicrobial. Okay, but importantly, only antimicrobial in its non-native range. One last thing that we did is we kind of pulled this into the biochemical world by getting extracts from a guy named Don Cipollini at Wright State in Ohio, who studied this plant and its chemistry for a long time. We've got a couple different fractions. This glucosinolate fraction, that's a classic mustard fraction. Flavonoids, which contain a chemical that's only really found in the genus, this Alliarianicide. And then we applied it to European and North American soils. Each one of these bars has multiple sites and multiple treatments. And spore viability of the spores being killed, well, yeah, some get killed in Europe in this experiment, but the effect is much, much stronger again in North American soil. So I'm just, again, trying to emphasize that we did find a really strong biogeographic difference in the way this plant affected this important component of the soil biota. One last feedback thing in this context is a paper that's in press right now, written by a colleague of mine named John Marin. And what this graph shows are feedback effects for a bunch of our grassland invaders, the ones that have a pretty big effect here in western Montana. Feedbacks on the y-axis, this is simplified so that a really strong negative bar means a really strong negative feedback. And if you're up near zero, nothing's going on at all. Black soil from the native range, that's Europe for all of these guys. And this is white is soil from the introduced range, that's us in Montana. In Montana, we just get nothing. 4 out of 6 show really strong negative feedbacks in their native range, showing you that a million times. But here's the weird thing about this graph, and something that's sort of an interesting take-home. All these species didn't exist or weren't present in the communities in Europe where the soil was collected. And so what it means is that we're getting kind of this controlling soil biota that's really widely spread, like at the scale of France or Germany or something, not at the scale of the particular meadow or at the scale of even the rhizospheres of the plants. And I would have never guessed this would have turned out this way. But this to our knowledge, this is brand new in terms of scale, and we're still scratching our heads and trying to figure out how this might work. But nevertheless, the bottom line was that all of these species were not present in Europe in the grasslands where these soils were collected. Their relatives were, but they weren't. Okay, one last little vignette, and I'll start wrapping this up. I did the arrow going down. Now I'm going to do the arrow going up. I'm going to leave biogeography because I don't have a biogeographical story here. But I want to show you a summary graph, and then give you a little story or vignette about some work that a student of mine did that's very unique in terms of the direction of this arrow. First, I want to just make an observation, and this is something we've kind of figured out in we ecologists have figured out in the last few years, and that is pretty consistent and powerful effects of invaders on ecosystem properties. I'm not really an ecosystem scientist, studier myself, but in terms of NPP, net primary productivity, one of the things that really we care about, in terms of ecosystem biology, across about 90 to 100 studies in a meta-analysis done by this guy, Liao, they find a pretty consistent increase, about a doubling in NPP. That's weird. Okay, so 83% across all of these studies, exotic invaders are increasing the productivity in their non-native range. Okay, and not only that, they're not only increasing productivity, they concomitantly increase the availability of a lot of resources, not decrease. That doesn't make sense. Well, intuitively it doesn't make sense. So I just put this over here to remind me, there's a big increase in nitrogen pools, okay, at least over the time frame of the decades of an invasion. And so this meta-analysis shows a lot of different examples of these sort of ecosystem properties that change with invasion. But the bottom line is you see an increase in NPP and you see an increase in the number of resources. And we're not really, remains a bit of a mystery. I have a student, Morgan Luce, who's defending this semester. She's tackled this question experimentally and spatially, just in terms of correlation, with 4 of our grassland invaders, and she finds the same thing. Except much more. She finds about a 2.5 times increase in NPP with invasion. And at the same time, she finds nitrogen pools skyrocketing, and she finds nitrogen availability skyrocketing, and some aspects of nitrogen flux increasing as well. And I'm not going to pretend I have an answer for that. But with that in mind, I'm going to go to a peculiar system and show you pretty much the same thing, and one kind of weird microbial reason for this. This was another PhD student of mine, Marnie Rout. She brought this problem to my lab. I didn't know anything about it. It's all of her stuff. I'm just reporting on it. This is sorghum halepense in the background. It's invading several meters a year in little blue stem prairie in Texas. And as it does so, it has some pretty big impacts. Sorghum halepense is weird. It's a relatively recent hybrid. I mean, thousands of years, but not millions. It's a relatively recent species. Okay, and when these two, one African and one Asian, European, I believe, sort of got together in the Middle East. They made a hybrid that was octoploid, and this thing has become invasive in different parts of the world, and including our southeast. Okay, it's also a biofuel plant. So the impact's pretty strong. This arrow should go the other way. Their species diversity goes way down. Productivity doubles. Okay, that above ground productivity. Over here we've got many species per meter squared. Over there we've got very few. Happens very quickly. Sorghum halepense drives them out, and she's measured this over time as it expands. She's also found that sorghum halepense does weird things to ecosystem processes. Back up. This is an ecosystem process. That's NPP, or close to it. But she's also found just exactly what I showed you before, which is this N is for native. This is nitrate, nitrogen, available nitrogen for plants. Low, this is transition. And then the invaded, high, 2 to 3 times as much in invaded systems. It's not sucking the nitrogen out of the soil, it's adding nitrogen to the soil. And the same thing here. This is for phosphate, and we see the same pattern. I don't know what's going on with phosphate, but we have a pretty cool hypothesis. She has a pretty cool hypothesis for this pattern with nitrogen. And it's because she found something really pretty novel. Sugarcane does this, by the way, but with different species. But what she found is that the rhizomes, which this plant makes a huge amount of rhizomes in the soil, that the rhizomes of sorghum halepense are loaded with nitrogen-fixing bacteria that can take N2 from air and make it into plant available nitrogen. They are housed inside, just like a nodule in a pea plant. They're housed inside the rhizomes of the plant and provide a lot of nitrogen to the plant. She has measured the nitrogen fixation rates of these rhizomes, and they're much higher than the typical reported for like a bean nodule. Very high rates of nitrogen fixation. She found that this nitrogen fixation was very important to the ecology of the plant. And again, I'm not going to show you graphs, but she figured out a way to use antibiotics to knock back the bacteria in the rhizomes. Plants died, rhizomes were tiny, sorghum lost its ability to compete with little bluestem. Sorghum is famous. A bunch of labs have studied the allelopathic effects of sorghum from the roots of this plant. Lost its allelopathic effect, and it couldn't defend itself against herbivores in her little petri dishes comparisons. So it appeared that this aspect, part of its soil biota, that this aspect of the microbial biota of this plant was very key to its ecological sort of success and its ecological sort of ability. So, last vignette with that. Keep this in. There are probably substantially other reasons for all of these other studies of invasives that show ecosystem changes, and we can talk about that afterwards. So I think this is it. My last slide. This is what I've covered: biogeographic differences in soil feedbacks. I think this is important for a couple of reasons. It brings microbial biogeography actually into the world of biogeography. There's clearly some biogeographic aspect to these critters. And it provides some kind of insight into exotic invasions. Maybe this is an important mechanism. Biogeographic differences in microbe-driven ecosystem processes. Talked about that a little bit too. And then this last bit about mutualists being harnessed by invaders. And we don't know yet about biogeographical effects of that aspect. Done. Thank you very much. I'll do my best to answer your questions.
So I can't remember the paper, but I saw somewhere where there's evidence that the invaders accumulate pathogens the longer they're here. So is that something that you've studied, and will that eventually sort of mitigate the effect of the invasion? Yeah,
the best paper was that I know of was by Power, one of the authors. It was a science paper. It was above ground pathogens, and I do think there's pretty good evidence that over time, and I think centuries, not millennia, that it's highly likely that pathogens that I've been talking about will evolve in response. I don't think the classic monocultures are around for millennia.
So I know it's a hard experiment, but does this make you an invader to have the positive feedback? And not being an invader means you don't have this positive feedback? I guess you could maybe tie that to agricultural crops that are moved on force
The
around
hardest
the world.
aspect about it, it's not necessarily that it would be hard to do, that it would be very dangerous to do. So in other words, all of the feedback stuff is done in really pretty controlled conditions. Many of the other hypotheses for invasion can be tested or explored in the field. In this particular case, I can't easily play with European soil biota in the field in Montana. So I would argue that all of these very strong or very weak negative feedbacks do correspond with being an invader, and I think it's a very good hypothesis that it makes you an invader. To escape from the biota that control you. Is that get your question right?
Yeah.
But couldn't you, it seems to me that the study design that would help figure this out would be to look at time sequence. In other words, have you had invaders that have been there for different lengths of time, and would you see the effect go away, basically?
Yeah.
Because you could get any range of invasion times, I guess.
Yeah, no, so there's two different lines of questioning going on here. I don't know of any examples of testing this idea of either lag time or disappearance time that involve this kind of soil biota effect.
Just by selecting different species, this is what I'm saying.
Yeah.
Invaders that have been there for different lengths of time.
Yeah, I don't know of any examples of that. I mean, there's a lot of reasons why that could happen. But for example, yeah, I see what you're saying. So for example, people have played with agricultural crops like corn, looking at either very, very new varieties and plant soil pathogen effects, or very, very old varieties and plant soil pathogens. There may be some way to do that. A guy that has done something, the garlic mustard example I gave you, there's a guy in Georgia, his name is Rick Langkow. And I'm not thinking quickly enough to come up with the perfect example, but he's got some really fantastic evolutionary sorts of history data suggesting kind of the waning or waxing of some of these effects. And I don't know if it's soil biota or not. That's about the only example that I can think of.
So you touched upon this briefly about herbivory, and I was thinking, this idea that if you're an invader, the soil biota would help you establish initially, but over time, what makes you a great invader isn't necessarily soil biota, but perhaps an escape from herbivory or other competition effects. So I guess, have you, the experiments where you transplant, do you do multiple generations to see if the invader...
2 generations. In other words, but these are generations, they're not really generations, they're 2 rounds of feedback experiments in a pot. And if I understood, I couldn't hear everything you said really clearly, but if I understood, yeah, there's no question that this isn't mutually exclusive of other ideas. But these guys have been around a long time, and we can collect knapweed that's been at least in the Bitterroot Valley for 50 years or so. And we still can find strong, very, very weak feedback effects on this plant. Well, very strong negative effects on natives. Okay. So it certainly could contribute to it being a strong invasion. Did I miss part of the question?
Yeah, sort of. We can talk more about this at lunch.
So if an organism moves out of its range, it's released. It could be released due to things in the soil.
Sure.
Entomologists have really
Sure.
enjoyed saying it's all entomology. So do you have a sense for the relative?
Well, I don't think biocontrol entomologists have ever said that it's all entomology. And I think it's in part, we've only really had a sense that the soil biota have this very powerful effect for less than a decade. Biocontrol work has been around for a lot longer than that. If you were to ask me the relative number of species, the proportional number of species, my brain is completely fading out, of papers that have focused in on soil effects, I would say I'm amazed at how consistent they are, the meta-analysis. If you were to ask the same thing about specialist insects, and remember, and a lot of these guys that are pathogens in the soil, they have their preferences, but there are very few of them that we know of that are truly like, one insect on one species of Centaria. These guys are rarely true, absolute specialists. They're kind of generalists that have their preferences, it appears. But the proportion of studies with specialist insects seems to be much, much lower in terms of really having this kind of powerful difference. But there are very few biogeographical studies because of that sort of thing of insects, because it's hard to do. So, and in terms of preferences for generalist insects, we found, I talked about this the last time I was here, we have found for knapweed really strong differences between generalists in Europe, which do just fine on spotted knapweed, and generalists in North America, which take it in the shorts when they're forced to eat spotted knapweed. So, yeah, so I'm not trying to discount other aspects like other consumers.
I was just trying to get relative feeling.
I'm just surprised at how often, how the very high proportion of studies of this nature that show strong soil effects in the context of invasion.
And this last example that you showed with this sorghum, I believe it was, and your student identified this bacteria that was providing nitrogen, did this sort of simple but elegant antibiotic kill experiment. Do those plants survive then when you supplement with nitrogen? Was it that simple?
Yeah, again, I could have shown, I was trying to keep the talk within about a 40-minute range. Yeah, she did. She had the antibiotic nitrogen supplementation. The nitrogen supplementation didn't do it all, but the nitrogen supplementation did dramatically kind of attenuate the effects of the antibiotic. That's a good question. Mark?
So what happens if you take an invader, let it go for a while, and then bring it back to its native range? Does it become a new invader?
No.
Is there competition between, if you do competition studies between invaders?
That's a great question. So that's a different kind of evolutionary question. In other words, somebody asked, well, you asked earlier, does that make it an invader? Maybe I misunderstood your question. So we're talking about evolution, which I haven't talked about at all, of the invader. There's a ton of evidence that invaders really rapidly evolve. Matter of fact, we've actually shown for Centaria solstitialis reproductive isolation between populations in the native and non-native range, which is pretty crazy after about 3 centuries.
So the genome sequence of the organisms are different then when it's not just their microbiomes are different.
Typically, the evidence for, yeah. So you'd have to go back and look at each one of these studies and have to think about it and look at the paper in terms of what we control for the actual plant source, the population source. But yeah, there's a ton of evidence that invasive species in their non-native range are beginning to have evolved in some typically ecological way. They compete better or worse. They're bigger or they're smaller. They're better defended or they're less defended. On and on and on. There's quite a bit of evidence. I don't have any evidence that indicates that makes them a better invader. Does that make sense? The way I put it? I don't have any example of a species heading up to North America, evolving a little bit, and going back to Europe and being an invader. I think by now something that would happen. So I suggest, I mean, the best working hypothesis, just like the soil, this is a community context kind of thing. And it's more complicated than just a change in a particular organism and how it interacts with other organisms that really matters. So, did that make sense, Mark?
Yeah, I'm just thinking about it in terms of, they've done these kind of experiments in microbial communities. They look at legacy over a long period of time, and you take something way out and then bring it back to the pathogens that were present in the beginning.
Yeah.
And it does great.
Yeah.
Again.
Yeah, and although that isn't really a total community context, I just don't know of any example. We're with a guy in Hungary, I believe, maybe I'm not immersed enough right now in the literature. We've done the reciprocal stuff with Solidago gigantea, which is a native here and then a bad invader in Europe. And we don't see any evidence in what we think is the first experiment of what you just asked. We haven't published it. We haven't seen any evidence that the Europeans come home and kick butt. They just don't.
We have 2 invaders here. Quapertensis, Kentucky bluegrass, and they're almost enormous, smooth
Yeah.
brome.
Yeah.
And talking to
A little
your
bit that
coach
before I had coffee.
years ago, said they would easily be
Rough.
classed as, what am I trying to say? As
Noxious.
noxious. But please don't ask anybody to do that. So I'm gonna compare those with your sorghum example. I think that POA is about twice as productive in fescue meadows as is the fescue community itself. And in the same meadows, clones of brome are about 5 times as productive. Exactly what you're saying there. And with that extra production comes extra competitiveness, so species diversity
Oh, yeah,
is pushed way down.
That's the classic model of invasion. Hundreds of